Hello, everyone. My name is Nathan Sprang, one of the organizers of the Feindel Virtual Brain and Mind Seminar. So why don't we get started today? Just as a reminder, if you could please mute your screen and audio during the duration of the talk, following which we'll have a moderated Q&A where you can type into the chat window or unmute yourself and ask questions. However, I'd ask that you hold questions to the end. So I'm very excited to have Alex Weissman joining us today. Due to COVID and the reason why we even have this virtual talk series, he hasn't yet joined us in Montreal, but he is an incoming postdoc with Sylvain Bellet. So Alex has had a quite extraordinary and ascendant career in academia. He completed his undergraduate degrees in both neuroscience and psychology at the University of Nebraska at Omaha. He also there completed his PhD in neuroscience under the direction of Tony Wilson. So in this very short period of time, Alex has accrued over 40 publications in peer-reviewed journals, top-tier neuroimaging ones, including Neuroimaging and Cerebral Cortex. So Alex's work is quite a wide range of activity, but really focuses on neurophysiological events measured with EEG and soon with MEG, looking at aspects of visual attention in aging and dementia. So I'm very excited to hear your talk today, Alex, on tracking the real-time encoding of item order information into memory.
All right, great. So thanks for the introduction, and thanks to everyone for tuning in to listen to my talk today. As was already said, my name is Alex Wiesman. I'm a freshly minted PhD from the University of Nebraska Medical Center, and I've spent the last 5 or so years using magnetoencephalography, or MEG, to study the neural dynamics serving cognition in health as well as in age-related disease. So I've primarily worked in HIV-associated neurocognitive disorders, and more recently in Alzheimer's disease. And I'm really excited to be continuing my training at the MNI with Stefan Baillet soon. But today I thought I'd go a different direction and go over some of the more recent work I've done, which leverages the high spatiotemporal resolution of MEG to study the real-time loading of item order memories into a progressive memory store in the healthy human brain. So to start, I'm planning to briefly touch on some terminology, but I won't spend too long on this because I figure that this crowd, if any, probably doesn't need much background on memory theory. I'll then dive into the need for dynamic encoding of item order information into episodic memory, as well as the growing recognition that multispectral patterns of population-level neural activity, called neural oscillations, are actually required for the human brain to support this process. I'll then show some results from my recent study, which aims to track the loading of item order information into cortical memory stores in real time across multiple frequencies of neural activity, before wrapping up with some hopefully persuasive reasons that I think these findings are important and how they might even be able to guide some future research in the field. So first, I want to focus on this idea that memory, and especially the episodic memory encoding, often needs to be relatively dynamic in nature and flexible. So when I refer to episodic memory, first of all, I'm meaning the subcomponent of autobiographical memory that consists of the events that happened and their temporal organization. So essentially, this is just what happened and when. And importantly, this is separable from the semantic relations that truly make these episodic details autobiographical in nature, meaning that any attempt to investigate this process shouldn't be confounded by personal connections to the memorized information, or at the very least should be sort of balanced across that kind of confound. And as I said, the process of encoding these events must be highly flexible and adaptable, since the temporal scale of important events in our environment is often pretty variable and relatively unpredictable in real life. And really, the encoding of these item order relationships with reasonable fidelity is of huge importance to us. So the example that I like to always give is that we've all been in the really unfortunate circumstance where you lose something extremely important to you, and you, after all other steps are exhausted, you eventually resort to trying to retrace what you did in the day since you knew you had that item, right? And this is a really frustrating process for a lot of people, but it's actually a really perfect example of how item order information can be used in our everyday lives to our benefit. So Very importantly, the flexible storage and retrieval of item order information is known to be degraded in a number of disorders affecting cognition, such as Alzheimer's disease and Parkinson's disease, both of which are of interest to me personally. But it also exhibits subtle but consistent declines across the course of healthy aging. So the study of the biological bases of this process will hopefully not only be informative for our basic understanding of the human brain and how it functions, but is also likely to provide some paths forward for therapy and possibly even diagnostics in a clinical setting. I've always found it really important to link even my more basic research to clinical utility. And at the end of the talk, time permitting, I'll discuss why I think this research in particular could potentially be beneficial in this way. So because of the importance of episodic memory for everyday function, its underlying neurophysiology has, perhaps unsurprisingly, been studied pretty extensively. In humans, this has primarily been done using PET and fMRI. And from these studies, a network of cortical regions have been established as being essential to episodic memory formation. So among these are the ventral prefrontal cortex, which in other types of studies has been really well established as being important for top-down regulation or executive control. So it's thought to do a very similar thing in episodic memory encoding. The ventral parietal cortex is also found in studies like this quite often. And this region of the brain is typically thought to be really important for attentional direction and orienting. So it's again thought to do something similar here. And then last but not least, and I'm sure I'm kind of preaching to the choir here, the medial temporal lobes have been identified as being really, really essential for the associative pairing of items that were previously considered unrelated in the context of episodic memory. So these studies, in concert with lesion and neuropsychology literature, have progressed our understanding of the neural system serving episodic memory hugely. But importantly, the vast majority of these studies have examined episodic memory on a time scale of seconds or even minutes, meaning that although they've provided really key evidence regarding the neuroanatomical basis of episodic memory, they do remain, at least in my mind, somewhat limited in their ability to experimentally parse apart the cognitive subfunctions and mechanisms that are involved in these processes. So at the best, most PET and fMRI studies of episodic memory have been able to dissociate encoding of information from its maintenance or its eventual retrieval, but each of these subcomponents are supported by a diverse set of cognitive mechanisms that, in my mind, remain somewhat unexplored. And over the past few decades, it's become increasingly apparent that many of the neural mechanisms I just mentioned are actually supported by what are called neural oscillations, which are large-scale population-level patterns of neural activity that are rhythmic in nature. Importantly, different speeds or frequencies of these oscillations, typically measured in cycles per second or hertz, are established as supporting divergent cognitive functions. And one of the most important findings from this research, in concert again with parallel lines of research, this time in animal models, is that the brain supports differing mechanisms of memory by organizing and transmitting information by means of these neural oscillations. So some of the most robust evidence for this oscillatory model of episodic memory comes in the form of theta gamma cross frequency interactions and their role in forming relational associations between, again, previously unrelated items. Now, this so-called cross-frequency coupling is represented by the diagram at the bottom of this slide and occurs when the phase of a slower oscillation, such as the darker sort of theta oscillation you see at the bottom of the slide, when the phase of that slower oscillation governs the amplitude of a much faster one. The faster oscillation here being represented by that sort of faster gamma amplitude that you see down there. Now, this process is thought to be a means by which information, again, is organized in the hippocampus as relational associations are formed during episodic memory. So again, in recent years, neuroimaging modalities with higher temporal resolution and sufficient spatial resolution have begun to answer the question of how exactly the human brain mechanistically supports real-time cognition. And thus, these studies have started to really delineate the rapid time-varying nature of memory encoding. So the figures you see here are from a particularly good example of this, where Heusser et al. From Lila Devacci's lab had participants encode streams of 36 nameable items, which you see on the top left there, separated into sets of 6, which is indicated by the color surrounding those items. And they hypothesized that if theta oscillations, if the phase of these theta oscillations does in fact temporally organize information in the hippocampus, then the progressive association of more and more items within a single temporal cycle, or in this case within a single theta phase, this should actually degrade the fidelity of these theta gamma interactions in this region. So colloquially, we all kind of know that 7 ± 2 rule for short-term memory. There's actually really interesting research that's come out recently showing that the speed of that theta phase can predict how many items a person is able to hold in short-term memory across people. So again, if that's kind of the concept here, you would expect that as you load more and more items into that single theta phase, you would then decrease the ability of that theta phase to sort of couple overlapping gamma responses that represent the individual stimulus items. So the model for that is shown in the middle left figure, and basically that's exactly what they found, right? They fit that model to the entire brain, and the results of that are on the statistical map in the bottom right. And you can see that where this model of progressive decrease in theta gamma coupling fit to the brain was the left medial temporal lobe pretty accurately. So this and other work has established a really key role for theta gamma coupling in the formation of temporal associations during episodic memory encoding, which was really what drove me into this line of research, because I found this study really fascinating and I wanted to see what I could do with similar kinds of tasks. But to me, this study left open a key question, which was, how are these associations between items and their order actually being held in temporary cognitive stores during the online encoding process? So in my mind, a few previous studies have actually explored this question, but they've done it relatively indirectly. And they've done this typically by investigating the encoding of information into short-term memory stores using magnetoencephalography. So a good example of this is shown here, where scientists actually from my PhD lab had participants perform a standard Sternberg style short-term memory task where they required participants to encode a series of 6 consonants, which you see sort of the task diagram is in the middle here. But they had participants encode a series of 6 consonants, hold this information in memory for a short delay period where it was just to be maintained, and then they had them indicate whether a target letter was a part of the previous set or whether it was not. Now, you might notice this task isn't inherently episodic in nature, and it actually doesn't require explicitly the encoding of any temporal relationships between these stimuli. But I think it's pretty well recognized in the literature that participants performing this type of a task, and especially when they're being tested with relatively high loads, as they are here, they tend to utilize a strategy where they repeat the items and load the items into an internal phonological loop. So basically, when you're trying to remember 6 letters in a row, you tend to read them left to right and then sort of encode them in this internal loop and repeat them in the same order, even though that order information isn't really essential for performance on the task. And I think interestingly, as you can see in the bottom panel, if you track the temporal progression of alpha activity, which is a little bit faster than theta activity, during encoding in this type of a task, it does appear to progressively recruit left-biased ventral parietal regions that are known to be really important for episodic memory, and it even starts to extend anteriorly towards the left inferior frontal regions that have also been implicated in this cognitive process. So basically, the takeaway from this is that left-biased alpha oscillations do seem to be important for short-term memory in some form, potentially for episodic encoding of item order memory information. However, a more exacting interpretation of this pattern of activity is really difficult in this kind of a task and with this kind of analysis. So in addition to encoding, alpha oscillations have also been shown to be really important for the active maintenance of visual information in memory. So in a really compelling line of research, Ole Jensen and colleagues, along with a few other laboratories, have shown that occipital alpha rhythms, which were originally really thought to just be the passive idling of the visual system, weren't supposed to be functionally significant in a lot of people's minds. But this work has shown that this occipital alpha rhythm is actually instrumental in the protection of maintained visual memories through the functional inhibition of incoming visual information that is distracting. And one of many of these studies is shown here. one that I actually really enjoy, where the researchers had participants perform, again, a standard Sternberg short-term memory task, very similar to the one that I showed previously, but this time with a little bit more order information intact, where they're showing the verbal letter stimuli in an order. But the main difference is that in this task, they actually presented either a weak or a strong distractor during the maintenance period of the task. So you can see that in the task figure on the left here. They either presented a weak distractor, which was irrelevant, just this bracket, or they provided a strong distractor when participants were supposed to be maintaining this information in memory. In this case, the strong distractor was another letter. So you could imagine this was more distracting because it could actually interfere with the stimuli that was being actively maintained. Now, what they found was that participants who had a stronger alpha synchronization in response to the strong distractor as compared to the weak distractor exhibited an enhanced ability to ignore the strong distractor and complete the task effectively. So in other words, participants who showed a stronger alpha synchronization were able to keep the memory representations intact more effectively and respond more effectively to the task. So to recap all of that, basically theta and gamma oscillations, and particularly the dynamic interactions between these spectrally specific responses, appear to be pretty essential for building associations between items within discrete time windows, and left bias alpha responses seem to generally be important for short-term memory encoding and maintenance, and potentially are important for episodic encoding of memory. Based on this, we predicted that these patterns of left bias alpha activity are actually a mechanism for the real-time loading of item order information into a temporary buffer or memory store. And we wanted to then test this more directly using MEG. So for the study I'll talk about today, we recruited 23 healthy young adults with a mean age of about 26 years and had each participant perform 120 trials of the task that I'll describe here in a moment. So during the task, we recorded participants' continuous neural activity using an Electa neuromag system with a sampling frequency of 1000 hertz, meaning that we had one data sample every millisecond, so pretty good temporal resolution here. And we then analyzed the resulting data from the 204 planar gradiometers that are embedded in our system. The task that we actually had them perform, which you can see at the bottom of the screen, was specifically designed for the purpose of the analysis that I'll describe in a few slides. But briefly, I'll describe the task. So each trial began with a fixation period, which was the fixation cross you see on the far left there. And this was randomly jittered to negate any anticipatory effects. After this fixation period was over, we presented them with a sequence of 3 novel semi-naturalistic objects called fribbles, which we drew from a database developed in Tom Barry's lab in Leuven. And after the sequence ended, participants had to maintain the item order relations between these novel objects in memory for a short delay period, for a short maintenance period, and were then probed with a forced choice regarding the order of the previously seen stimuli. So you can see examples of that on the far right here, where on the top there was a non-intact item order trial, and on the bottom there was an intact item order trial. But since we were focusing in this analysis on the encoding of this information, we kind of collapsed those two types together and really focused on the encoding data specifically. And one more thing that's really important to note here is that all of the 306 stimuli that the participants saw over the course of the task were unique to them. So, or at least were unique over the course of the trials. So we didn't have any repeats across the entire course of the trial of the study, which I think is a really important point to make here. Now, our first step in terms of data analysis for this type of a task is usually to identify the time and frequency extents of significant neural responses to the sequence items at the level of our sensor recording. So just taking a step back, in our laboratory at least, we think that it's really important to define the time and frequency constraints of these neural oscillatory responses to stimuli on a group by group and a study by study basis. The reason for this is we've actually found that across different age groups, across different patient populations, and even across different tasks, so whether you're doing selective attention task or sort of a more basic visual task, the time and frequency definitions of these responses do seem to systematically change in a lot of meaningful ways. So The way we get around that is we actually identify these on a study-by-study basis rather than just assuming their time and frequency extents, although they are relatively consistent across studies. So the spectrogram that you see presented centrally on this slide is a grand average from across all participants at a representative sensor that's over lateral occipital cortices. And as you can see, time is displayed there in milliseconds on the x-axis, and frequency is displayed in hertz, or again, cycles per second on the y-axis. And each of the dotted lines that you see overlaid denote the onset of a new subphase of the task. So the onset of a new sequence item or the onset of the maintenance period. And as you can see, the onset of each sequence item induced a robust response in the alpha range, extending from roughly 8 to 14 hertz, and extending for about 1,000 milliseconds in every case. Now, you might have noticed that these responses are either blue or red, and I haven't really described that yet. But this means that they vary in their direction relative to the pre-stimulus baseline period. And I think this is a really good time to note that the direction of these responses relative to baseline is actually very important. So since we're measuring levels of rhythmic synchrony relative to, again, a pre-stimulus baseline period, decreases in synchrony, or desynchronizations as we call them, are equally important to increases in synchrony. And in fact, alpha and beta desynchronization responses are the ones that have been shown to be the oscillatory neural responses that are most collinear with fMRI bold activations. So basically all of that was just to say that these desynchronization responses are, at least to us, at least as interesting as their inverse. So we used cluster-based permutation testing to then identify the significant time frequency windows that contained our neural oscillatory responses of interest. And you can see the spatial distribution of these responses at the sensor level in the bottom panel of this figure. And I think it's important to note that even at this relatively coarse level of analysis, you can already see that there appears to be a progressive recruitment of more anterior cortices along the left side of the brain as the memory sequence progresses. So as we go from the first sequence item to the second to the third, you can see there's sort of this progressive recruitment of left lateralized anterior cortex, which I found very interesting. So once we knew the time and frequency definitions of our oscillatory neural responses to our sequence stimuli, our next step was that we wanted to model these responses at the level of the cortex. And we did this using a frequency resolved beamformer called the DICS beamformer. And you can see the results of this analysis on this slide. So first, to orient, these maps are again grand averaged across all participants per each sequence item. And again, as more item order information was progressively loaded into memory stores, you can very clearly see that more anterior cortices were being recruited by this alpha frequency desynchronization response. Now, this is obviously great that we can see it again at this level, but we of course actually wanted to test which regions were exhibiting this progressive loading effect statistically rather than just looking at it qualitatively. And the design of our sequence memory task actually allowed us to use a pretty intuitive slope printing analysis for this. So I'll describe that now. So due to the timing of our item presentation, we were actually able to image the neural response to each item in the sequence individually, even though they were occurring in pretty rapid temporal succession. And then using these item-specific images, we were able to model a slope that represented the progressive encoding of item order information at each voxel in the brain in real time. So for a synchronization response, the interpretation of this analysis is pretty straightforward, where a progressive increase in the amplitude of the neural response would be represented by a steeper positive slope, which would then be interpreted as a more robust item order memory load effect. In contrast, a more shallow slope would mean that there was a less robust load effect in that region. But in contrast, and of particular importance to our analysis here, because we're looking at an alpha response that is a desynchronization, meaning that it's negative going, any negative going responses would be interpreted inversely from positive sign waves. So a steeper negative slope would indicate a more robust item order loading effect, while a more shallow negative slope would then also, again, kind of indicate a less robust loading effect here. And again, it's important to really reinforce this idea that this slope was modeled at every single voxel of our volumetric brain space. So we were able to take this slope, this voxel-wise slope data per participant, test this against a null hypothesis of 0, which would indicate no sort of uniform encoding effect in that voxel, and then use that to determine which regions did seem to exhibit a significant item order loading effect. So the map that you see in the center of this figure is the result of that analysis in the alpha band for those desynchronization responses, where again, just to reinforce this, a slope was modeled at every single voxel across the 3 sequence items, in this case just using MATLAB's polyfit function. These voxel-wise slopes were tested against the null hypothesis of 0 across all participants, and these maps were then corrected for spatial multiple comparisons using a relatively stringent FWE procedure. And consistent with our previous qualitative observations, this analysis revealed a strong progressive item order encoding effect in a network of left biased cortical regions, all of which were pretty intuitive and pretty exciting to see sort of fall out of this analysis. So among these regions were the ventral dorsolateral prefrontal cortex, the ventral parietal cortex, and the superior temporal cortex, all of which are kind of already associated with episodic memory encoding. But hopefully in this case, we're actually providing a little more detail of what these regions are actually doing. So each of the violin plots you can see projected from these subpeaks shows the density of the participant level data per each sequence item. And the gray lines overlaid on these plots show the single subject slopes centered on their own mean. So I think, again, it's really kind of cool to see that even at the level of each single participant, there seems to be pretty strong evidence for a progressive negative slope across these regions, indicating a progressive loading effect. So that was all really interesting, but obviously we then want to make sure that we're connecting this directly to performance on the task, right? Do these dynamics actually seem to relate to item order memory performance on our task? So to do this, we extracted alpha frequency time series data from each of the 3 subpeaks that I just talked about per every trial for each participant. And we use these data to again fit linear slopes to again, in this case, every single trial at those regions in the alpha band. And we fed these data into a linear mixed effects model to predict trial-wise reaction time with participant and trial as nested random effects. And all of this was done using the NLME package in R. And you can see the actual model sort of spelled out at the bottom of this slide. So the plot you see here has the alpha encoding slope, again indicating that progressive loading effect, in this case in the ventral parietal cortex. But the x-axis shows this encoding slope per trial, as well as reaction time per trial on the y-axis. And each of the fit lines and shaded confidence intervals that you see overlaid are indicating a different individual participant. And as you can see, there was a modest but significant relationship between these variables, such that on trials where participants exhibited a stronger item order encoding effect, meaning a more negative slope, they also responded more quickly regarding the item order relationships in the sequence. So this was really nice because at least in ventral parietal cortex, we actually found a direct link between this progressive encoding effect and performance on the item order regarding the temporal relations, the item order relationships between our stimuli. Now, since again, we're testing the real-time loading of item order information into a memory store in this task, we wanted to next see whether this encoding slope effect actually impacted neural activity during the phase of the task where participants were required to actually keep this item order information in an active memory store, right? So that would be the maintenance phase of the task where no items were present and they were just sort of holding this activity in memory. So to do this, we again used a linear mixed effects model where we modeled at the single trial level the encoding slope at each of these regions in the alpha band. And we also modeled the neural activity present in that same region during the maintenance period that followed. And what we found was that as the encoding slope effect was stronger or more negative, participants seemed to exhibit a more robust response during the maintenance phase of the task. So this really indicated that the relative loading of item order information during encoding was directly predictive of the amplitude of neural activity in that same region on a trial-wise basis, while that information was actually actively being maintained for later retrieval. So up until this point, I've been focusing on the encoding slope findings in the left hemisphere, so that frontal, parietal, and temporal sort of network of regions that we found. And this is mainly because I find these the most interesting, honestly. But there was also a really interesting cluster that I want to touch on that was in the occipital cortices, that although it didn't survive our relatively stringent correction for multiple comparisons, I think could give us a little bit of information of what's happening in the brains of these participants over the course of the encoding of these progressive item order relationships. So as you can see from the statistical map displayed here, the affected visual cortex was actually the opposite of what we found in the left frontal, parietal, and temporal cortices, such that as the memory sequence progressed, the desynchronization response in this region actually decreased in amplitude. So there was less of a response as more information was being encoded. And to us, this was really interesting because what it signifies is that there's likely a separate process that's occurring in these earlier sensory cortices, which was functionally distinct in some meaningful way from what was happening in these left-lateralized regions that showed more of a progressive encoding effect. So to dive into this a little bit more, and as a final analysis for these alpha responses, we then use the three subpeaks from those left bias slope regions. Again, so the ventral prefrontal, dorsolateral prefrontal, the ventral parietal, and the superior temporal cortices. We used each of those three subpeaks as a seed and used them to compute spectrally resolved coherence with every other voxel in the brain using a DICS coherence beamformer. So the statistical maps you see underneath each seed region on this figure are the only clusters from this analysis that survived our correction for amplitude confounds, which is something that's actually very important to correct for in this kind of analysis in MEG. But they also survived our cluster correction. And as you can see, the activity of left hemispheric regions that appeared, again, to index this progressive encoding of item order information, the activity in these regions was at least partially functionally decoupled from earlier visual association areas relative to the baseline period. So again, important to note here that this is all relative to a baseline period. So the voxel-wise data you're seeing in these statistical maps in the bottom is actually the amount of connectivity or coherence between these regions relative to the coherence that was present at baseline. So since I have a little bit of extra time, I'm gonna now touch on some preliminary findings in another frequency band that I think are also really interesting here. So when I showed this figure earlier, you might have noticed that in the spectrograms, there's actually a pretty consistent lower frequency synchronization in the theta band, as well as that desynchronization you see in the higher frequency alpha band. And if I show the spatial topography of these responses, you can actually see that, again, there's an early indication of a potential sort of stepwise change in the amplitude of these responses as a function of the progression of that sequence encoding. And when we image these responses to the cortex and perform the same encoding slope analysis on them, we found that they exhibited a negative slope across bilateral visual cortices, as well as in the right medial temporal lobes, meaning that the amplitude of this response, again, actually decreased as a function of item order load, or as a function of the progression of those sequence memory items, in a really similar pattern that we found in the alpha oscillations in primary visual cortices. So again, this is preliminary and is somewhat difficult to interpret, honestly, because it likely also indexes some aspects of alertness or even stimulus novelty at the onset of each trial. But I still find it really interesting that visual responses generally showed an effect that was inversely related to the effects that we saw in the left hemispheric frontal and parietal cortices during the active encoding of these item order relationships. So the main conclusions that we can draw from these data are that left hemispheric networks of regions oscillating specifically in the alpha band appear to index the real-time encoding of item order information into memory, and as would be expected, the magnitude of this encoding effect predicted both neural activity in the same regions during maintenance, sort of indicating this active loading into a memory store. But they also predicted the item order memory performance on the task, sort of linking this directly to the participant's ability to actually recall the item order relationships in question. Now, we think that these findings might provide a more specific explanation for a number of previous studies of short-term memory, which again reported a gradual progression of alpha desynchronization from early visual to left frontal and parietal cortices as the encoding of items progressed. But importantly, this seems to distinguish those responses a little bit more effectively than previous studies. And I think finally, one thing that's really important to take away from this is that this progressive encoding effect was functionally distinct, not only from patterns of neural activity in primary visual cortices, but also in more lateral occipital visual association areas, which I think really shows that this is potentially sort of a distinct network of regions than sort of just the visual networks that are helping to encode these items in kind of a different way. So as promised, I'll talk a little bit about where I think this research, what this research informs and where it could potentially go in the future. I think most importantly, and something I always try to drive home with a lot of the research that I do, is that especially when we're looking at cognitive deficiencies in specific diseases, so things like Alzheimer's disease, we typically start with neuropsychological testing, for instance, and say that, for instance, people with Alzheimer's disease have issues with episodic memory encoding itself, right? But there's actually a lot of cognitive sub-mechanisms that seem to be really important for the encoding of memories into episodic memory stores or things like that. And I think that with more temporally precise measures like this, we're able to actually piece apart those mechanisms a little bit more accurately, and actually then use that information to hopefully guide both clinical outcomes for therapeutic interventions, or maybe even actually guide therapeutic interventions a little bit more effectively, which leads me into my next point really well, which is that potentially we could use this kind of information to enhance targeted neuromodulation therapies with the frequency-specific information that we're getting from these kinds of studies. So there's a lot of new research that's really focused on rhythmic TMS, or even transcranial alternating current stimulation. So both of these methods are non-invasive neuromodulation therapies that are able to actually alter the sort of extracellular environment and the neural excitability of groups of neurons at very specific frequencies, right? So both of these are rhythmic in nature. They have kind of different mechanisms behind them, but they're both basically inducing rhythmic neural activity, or at least changes in the neural environment at a specific rhythm. Now, one of the main drawbacks of these methods that a lot of people have recognized is that they're spatially pretty non-specific. So you're targeting generally large areas of cortex, and the obvious caveat, the obvious drawback there is that these brain regions are not important for only one cognitive function, right? So if you're targeting, you know, the ventral parietal cortex with one of these methods, and sort of doing it in a non-specific way, you're probably affecting more than just episodic memory. You're probably affecting more than just the specific cognitive function you're trying to target. So potentially, by understanding the sort of rhythmic properties of some of these neural activities, what we can do is we can actually target these therapies not only spatially but also functionally, where we can say we know that this specific function, this specific region is functioning in the alpha band for this specific kind of cognitive faculty. Maybe we can target that specific frequency of activity and try to make this a little bit more focal to specific cognitive sub-processes that we're interested in. So finally, one thing that I think is important to note also is that this is really a jumping off point for me. I think that this task produced some interesting results, but I think that it's always really interesting in my mind, especially when we're looking at sort of adapting these kinds of findings to clinical populations, to interpret them with a grain of salt, right? Because all of these studies are done in a vacuum, right? We're specifically targeting episodic memory encoding of item order information with this kind of a task. But in the real world and in situations that are relevant for these clinical populations, cognition never happens in a vacuum, right? So I think it'd be really interesting with this kind of a task to look for interactions between, for instance, this progressive episodic memory effect, so between that and maybe attention, so I've studied attention a lot during my PhD, and I always think that it's important to look at whether attention might interact in really meaningful ways with some of the cognitive faculties we're looking at. But I think even more importantly, it's important to look at how the properties of the actual items being memorized or being sort of encoded into a memory store, how the properties of the stimuli might also interact with this process. So is there an effect of semantic congruence on this kind of process? Is there an effect of the emotional valence of the stimuli that are being encoded? I think these and other questions are really important to sort of systematically parse apart how this kind of cognition is more likely to function in real world situations rather than sitting in a dark room in front of a screen with your head in a scanner not being able to move. So with that, I want to thank everyone for listening. Before I stop talking finally, I want to thank everyone who's been really important in this research. So first and foremost, my PhD advisor, Tony Wilson, was incredibly supportive of really all of the lines of research that I chased during my PhD, whether or not they ended up working, which was really nice because it gave me some freedom to explore some more interesting but also risky questions, I think. I also just need to thank everyone in the Conda Lab who are all incredibly supportive and really taught me a lot during my time at UNMC. In particular, in this case, Nick Christopher Hayes, whose picture you can see on the right there, was really sort of essential in some of the late stage analyses for this project. So special thanks go out to him. And then finally, I need to thank the funders as always. Believe it or not, this project specifically was supported by a research fellowship from NASA. So a lot of thanks go out to them for investing in this research, as well as a lot of the data recordings and the analysis equipment that were used for this project were supported by the NSF and the NIH. So obviously always important to have good funding for this kind of research, and great thanks go out to them. So with that, I'm done talking and I can take any questions.
All right, great. Alex, thanks so much for sharing that work with us. So that's right, we'll open up the chat window for questions, or if anyone would like to just unmute themselves, they're welcome to. So I'll just kick off here. So I thought it was really interesting, some of the source localization you were doing with the MEG data.
Yeah, absolutely.
Particular, with your encoding slope analysis, it seemed like, some of the parietal findings are convergent with, the fMRI findings. But I'm wondering, some of these other regions don't seem so consistent with like the earlier bold studies that you showed for encoding.
Yeah, especially that superior temporal cortex, right? That one's kind of.
Yeah. So I'm wondering, how does this converge with fMRI methods, particularly like across modalities?
Yeah, absolutely. That's a great question. I mean, so first of all, the obvious caveat is that MEG has a better temporal resolution, but you do trade off some spatial resolution there. So every finding that I have in MEG, I kind of, I say ventral parietal cortex, right? But not really. I don't try to interpret specific gyri as much as would be probably possible with fMRI or things like that. I do think the superior temporal cortex is a little bit more difficult for this one. I wonder if, in the back of my head, especially for this task, these objects were novel, and they were kind of naturalistic looking. And originally, I was looking for stimuli that were unnameable, if possible. But that is a surprisingly difficult task to find unnameable stimuli, right? Because when you tell people to do a task like this, that's not necessarily extremely difficult, but not easy either. Of memorizing these items, they tend to verbalize them whether or not you want them to, right? So in my mind, the superior temporal cortex, pretty much right where the subpeak falls right there, there's a lot of interesting research that I've seen in EEG and fMRI that link that to sort of multisensory integration. So to me, there's a good chance what's actually happening there is that's more just the sort of progressive associating of these visual stimuli with a verbal construct of some kind. But again, that's totally tentative. I think that, this is maybe a step towards parsing apart some of these subfunctions a little bit better, but it's not perfect. And I think that's that peak definitely required some reverse inference on my part, because I didn't know exactly how that aligned with fMRI research.
All right, great, thank you. So do we have any other questions from the audience? All right, Alex, why don't we go ahead and wrap it up for today?
Okay, perfect.
Thank you so much for coming and for sharing your work. And
Yeah, happy to.
we look forward to seeing a lot more of it in your time at the Neuro over the next few years.
Yeah, I'm really excited for it.
All right, great. Well, thanks so much, and we'll talk soon.
Yeah, thanks so much for inviting me.
All right, thanks, everyone.